Key to this momentous leap in understanding were technical developments in microscopes made by the Dutch lens maker Antony van Leeuwenhoek in the 17th century. He was the first to visualize microbes, but it was not until the mid-1800s that Louis Pasteur, working in Paris, and Robert Koch in Berlin established germs as the cause of infectious diseases. Pasteur was instrumental in dispelling the general belief in spontaneous generation, that is, the generation of life from inorganic material. He showed that mold growth could be prevented in broth by boiling and then placing it in a chamber with filters to exclude the entry of any particulate material from the air. This demonstrated the existence of airborne microscopic germs. Koch discovered the first bacterium, Bacillus anthraci, in 1876. He soon developed methods for growing microbes in the laboratory, and then, one after another, the causative microbes of feared diseases like anthrax, tuberculosis, cholera, diphtheria, tetanus, and syphilis were identified and characterized. It became clear that bacteria have a structure similar to mammalian cells, with a cell wall surrounding cytoplasm that contains a single coiled circular molecule of DNA. The majority are free-living and can manufacture all the proteins they need to metabolize and divide. However, there remained a group of infectious diseases for which causative organisms could not be found, such as smallpox, measles, mumps, rubella, and flu. These microbes were obviously very small as they passed through filters that trapped bacteria and in consequence were called filtrable agents. At the time, most scientists thought these were just tiny bacteria. In 1876, Adolf Meyer, director of the Agricultural Experimental Station in Wageningen, Holland, began to investigate a new disease of tobacco plants which devastated the Dutch tobacco industry. He called it tobacco mosaic disease because of the mottled pattern it produces on the diseased plant's leaves. He showed that the disease was infectious by transmitting it to healthy plants using sap extracted from a diseased one. He thought that the disease was caused by a very small bacterium or a toxin. Later, biologist Dmitry Ivanovsky also worked on tobacco mosaic disease at the University of St. Petersburg in Russia, and in 1892 demonstrated that its causative agent passed through filters that trapped bacteria. Like Meyer, he suggested that it was caused by a chemical toxin produced by a bacterium. In 1898, Martinus Beijerinck from the Agricultural School in Wageningen followed up on Meyer's experiments by showing that the agent grew in dividing cells and regained its full strength each time it infected a plant. He concluded a living microbe was responsible and was the first to coin the name virus, from the Latin meaning a poison, venom, or slimy fluid. By the beginning of the 20th century, viruses were defined as a group of microbes that were infectious, filtrable, and required living cells for their propagation, but the nature of their structure remained a mystery. In the 1930s, tobacco mosaic virus was obtained in crystalline form, suggesting that viruses were purely composed of protein, but shortly afterwards a nucleic acid component was discovered and shown to be essential for infectivity. However, it was not until the invention of the electron microscope in 1939 that viruses were first visualized and their structure elucidated, showing them to be a unique class of microbes. Viruses are not cells, but particles. They consist of a protein coat which surrounds and protects their genetic material, or, as the famous immunologist Sir Peter Medawar termed it, a piece of bad news wrapped up in protein. The whole structure is called a virion, and the outer coat is called